Hello everyone, I am Aniket Dattatreya. Today I am talking about antigen and antibody. So let's start. When we are studying about antigen and antibody, one question is arised: what is meant by antigen? So let's start. Antigen is nothing but the molecule that initiates the production of an antibody and causes an immune response. In that, antigens are large molecules of proteins present on the surface of the pathogen, such as bacteria, fungi, viruses, and other foreign particles. When these harmful agents enter the body, it induces an immune response in the body for the production of antibodies. For example, when a common cold virus enters the body, it causes the body to produce antibodies to prevent from getting sick. Next point is properties of antigens. There are various properties of antigens like the antigen should be foreign substance to induce an immune response. Second one is the antigens have a molecular mass of 14,000 to 6 lakh Dalton. Third is they are mainly proteins and polysaccharides. Fourth one is the more chemically complex they are, the more immunogenic they will be. Fifth one is antigens are species specific. And sixth one is the age influences the immunogenicity. Very young and very old people exhibit very low immunogenicity. Next point is types of antigens. First is on the basis of origin. There are different types of antigens on the basis of origin. First one is exogenous antigens, second one is endogenous antigens, third one is autoantigens, fourth one is tumor antigens, and fifth one is native antigens. In exogenous antigens are the external antigens that enter the body from outside. Example, inhalation, injection, etc. These include food, allergen, pollen, aerosols, etc. and are the most common type of antigens. Second one is endogenous antigens. In that endogenous antigens, they are generated inside the body due to viral or bacterial infection or cellular metabolism. In autoantigens are the self proteins or nucleic acids that due to some genetic or environmental alterations get attacked by their own immune system causing autoimmune diseases. Next one is tumor antigens. In the tumor antigens, it is an antigenic substance present on the surface of tumor cells that induces an immune response in the host. Example, MHC1 and MHC2. Many tumors develop a mechanism to evade the immune system of the body. Next one is native antigens. In the native antigens, an antigen that is not yet processed by an antigen presenting cell is known as native antigens. Next one is on the basis of immune response. On the basis of the immune response, antigens can be classified as immunogen, heptane. In immunogen, these may be proteins or polysaccharides and can generate an immune response on their own. Next one is heptane. These are non-protein foreign substances that require a carrier molecule to induce an immune response. Next point is structure of antigens. In the structure of antigens, the epitopes or antigenic determinants are the components of antigen. Every antigen has several epitopes. An antibody has at least two binding sites that can bind to specific epitopes on antigens. In that, the antigens combine with the antibody according to the lock and key mechanism. In that, the ability of the body to act against the disease causing agents and antigens by the immune system is termed as the immunity. This immunity may be either inborn or acquired from vaccinations. Second point is antibody. In that, ever wondered why your neck region gets swollen when you are down with cold and flu? This is because WBCs accumulate around that region, which is our body's defense mechanism in the blood. In that, antibodies are not found at a place as such, but whenever our immune system encounters antigen or pathogen, B cells get activated immediately, releasing antibodies into the bloodstream. These immunoglobulins undergo mitosis, resulting in cell division, and continuously produce antibodies as a result of producing more cells. These antibodies remain in the blood for some time But B cells remember these antigens and repeat the same course of action whenever they appear in our body. Next point is, what are antibodies? Antibodies is nothing but the immunoglobulin. These are large Y-shaped blood proteins produced by plasma cells. They bind to foreign particles and invade them. These particles are foreign bodies that get attacked by antibody. That antigens are foreign pathogens that invade the body and have the capability to give rise to response from our immunity system, either by grouping up with a larger molecule or alone after binding with antibodies for a particular immune response. Hence, antigens stimulate the production of antibodies by the immune system. Next point is structure of antibody. In the structure of antibody, an antibody has a Y-shaped structure made up of four polypeptide subunits. Each subunit has two identical light and heavy chains. In that, the N terminus of each heavy chain forms an antigen binding domain with a light chain. There are two antigen binding domains forming the arms of the Y shape. They are known as fragment antigen binding, that means FAB domain. In that, the C terminus of the heavy chains forms fragment crystallization, that means FC domain, which helps in the interaction with the effector cells. All four polypeptide subunits are held together by disulfide and non-covalent bonds. In that, the heavy chains of the antibodies contains a variable region and three constant regions. Each antibody has two identical antigen binding sites and they differ in the antibodies. Next point is types of antibodies. In the types of antibodies, antibodies or immunoglobulins, that means Ig, are of five different isotypes. This classification is on the basis of their H chains. Let's look at the different types of immunoglobulins and their functions. First one is IgM, that means immunoglobulin M. IgM is the first antibody produced in response to a microbial attack by B cells. That it is the largest antibody and is found in a pentameric form. In that, it circulates in the blood and lymph and constitutes 6% of the total antibody content in the serum. It is involved in agglutination and opsonization. In that, it has a large number of antigenic sites on its surface and therefore facilitates efficient activation of the immune system. Second one is IgG, most abundant isotypes in the plasma and comprises 80% of the total antibody content in the serum. It detoxifies substances that are harmful and recognizes the antibody antigen complex. In that, it is transferred to the placenta through the fetus and protects the infant until its birth. In that, IgG is divided into four subclasses, that is IgG1, IgG2, IgG3 and IgG4. Among these, only IgG3 and IgG4 possesses the ability to cross the placenta. In that, the heavy chains of IgG have two antigen binding sites and are of the subclass gamma. It facilitates the process of phagocytosis and provides immunity to the developing fetus. It neutralizes the toxins and pathogens and offers protection to the body. Next one is the IgA. In that IgA, usually found in liquids such as breast milk, serum, saliva, fluids of the intestine. In that IgA in breast milk protects an infant's gastrointestinal tract from microbial activity. It constitutes 13% of the total antibody content in the serum and is divided into two subclasses, that means IgA1 and IgA2. Among these, IgA1 is highly found in the secretions and is also called the secretory immunoglobulin. It exists in both monomeric as well as the dimeric forms. That means it provides the first line of defense against the pathogens and limits inflammation. It also activates the complement pathway and participates in the immune response. Next one is IgD. In that IgD, it is involved in the production of the antibody by B cells. It is present as a monomer and weighs around 1,80,000 Dalton. It comprises less than 1% of the total antibody content in serum. In that, it acts as a receptor on B cell surface and participates in B cell activation and differentiation. Next one is IgE. is that IgE is present in the least amounts around 0.02% of the antibody content in the serum. In that these are present in the lining of the respiratory and intestinal tract and respond to allergic reactions. This is found as a monomer in the body and weighs about 2 lakh Dalton. Next point is function of antibody. There are very very functions of antibodies like binds to pathogens. It activates the immune system in case of bacterial pathogens, directly attacks viral pathogens. It assists in phagocytosis. Antibody provides long-term protection against pathogens because it persists for years after the presence of the antigen. It neutralizes the bacterial toxins and binds the antigen to enhance its efficiency. They also act as the first line of defense for mucosal surfaces. They ingest cells by phagocytosis. In that, few antibodies bind the antigen present on the pathogens. These aggregates the pathogen and they remain in secretions. When the secretion is expelled out, the antigen is also expelled. These are all about antibody and antigen. Thank you.